Okay, the second lecture about fasting, we are going to be looking at some morphological and physiological changes associated with it. Essentially, a reminder of some basic information. How does intestinal remodeling occur in mammals? Intestinal remodeling in mammals occurs by cell proliferation that is continuous, actually replacing the cells that have died by apoptosis. New cells, the new cells that are proliferating in the crypts of Linköping down here, basically start migrating down to the tip of the villi. And here, up here in the tip of the villi is where cells basically will go apoptosis, will die, and will be discarded of. So what you have is a wave of cell replacement. Cells that are by mitosis produced here in the crypts of Linköping basically will end up migrating until they die. And this is a constant flux. There is a constant flux of mitosis taking place here, replacing the cells that are dying higher up in the villi. And there is a high replacement of cells here. These cells, the ones of the epithelial mucosa here, are actually called enterocytes. And as I said, they are born, if you want. They proliferate, they are divided by mitosis here, and they die up here by apoptosis. Cell proliferation, in a normal situation, you have a balance between how many cells are actually produced and how many cells are actually replaced, or how many cells undergo apoptosis. And what we do know is that the proliferation is inversely related to the activity of the gut. The more activity, that makes little sense. I have to look it up. It should be the opposite. The more activity of the gut, the more proliferation. That would be my expectation. Let's look at that from the point of view of the plasticity of the gastrointestinal tract. First of all, this is actually a clinical problem. We are not only talking about animals. There are many patients in hospitals that cannot feed by themselves, and they are fed using parenteral or enteral feeding. Parenteral feeding basically involves the administration of a nutritional formula in the veins, containing glucose, amino acids, lipids, vitamins, and added minerals, bypassing digestion. So you have a total bypass of digestion. In this case, these patients basically receive the nutrients directly into the blood. The gut is not being used. What would be the consequence of that? The atrophy of the gastrointestinal tract. Enteral feeding basically involves the administration of nutrients through a feeding tube directly in the stomach. And this is actually preferred. It's more cumbersome, but it's preferred. Why? Because in this case, you keep actually the gastrointestinal tract working, doing something. So, if you can avoid parenteral feeding, you do it by enteral feeding as a much better strategy to avoid actually the degradation of the gut. In this case, the alterations again, of course, involve cell proliferation and cell shedding. And we have an example of this. This is an example in rats. And in rats, what we can see is actually structural changes that occur when these animals have been fasted for, I think it was 2 days. Does it say anywhere? I think it's 2 days. So basically, what do you see here? You see, in this part, in the A part, you have control rats. This would be the structure of an intact villi. LP stands for the lamina propria. That's the basal membrane over which the villi is constructed. And these are the enterocytes. These are the cells of this epithelium that actually, as I was telling you, migrate up to the villi. What do you notice? What is the main difference between this and actually this picture here? This picture is in rats that have been fasted for 2 days. What you see is a more loose structure. The lamina propria is actually retracting. Instead of being close to the tip, is retracting down. You see that here as well. You see more vacuolization. Vacuolization as a first sign of cell death, of apoptosis. So there is an increased degradation of cells here that you don't see here. That's only after 2 days of fasting in rats. What you see as well, this is another picture here, a reduction of the mucosal cell renewal and the cell migration rates from the crypts of Linköping. control rats, fasted rats. And these images correspond to the section of the villi. In the previous slide, you had a section this way. Now you have a cross section. So you see the villi as these round structures. Notice the presence of fewer villi, the presence of some in which there is no lining appropriate, so they are basically being atrophied all along. That's not the case here. So you have a shortening as well of the villi and a decreased jejunal mucosal mass by 1/2, only after 2 days. So it is a large change, morphological change that occurs in rats. How does this look like in naturally fasting species? Sorry. In naturally fasting species that actually fast, you have an alternance between anorexic and hyperphagic periods. The thing is that in mammals, ourselves, how many hours go between meals? 3, 4, 5, up to 8 hours? It becomes a cycle. But of course, for animals that are naturally fasting, this can be a much longer period. And then there is an alternative of this anorexic period, no eating, fasting, hyperphagic period, a period in which you have to eat a lot to compensate for when you have not been eating. In penguins, you have this, anorexia on land and hyperphagia at sea. Penguins don't eat on land, so when they are there, they are not eating. In penguins, basically, you have these short fasts that last between 1 and 3 days when the animals are small. Here you have the smallest penguin, the little blue penguin. Basically, will have fasts that will last between 1 or 3 days. This penguin cannot fast much longer than that. Middle-sized penguins, the Adelie penguins, for example, up to 2 weeks of fasting. The emperor penguin, the champion, 4 months. The male emperor penguin incubates the egg for 4 months. During this period, the animal is not eating. So, of course, size has something to do with these responses with natural fasting. Well, and here now I'm introducing you August Krog. August Krog has been called the father of comparative physiology. And August Krog basically would justify why study penguins. In his words, we should study penguins because there is always a large number of problems for which there is an animal that is especially suited for that study. August Krog actually received the Nobel Prize in 1920-something. He was a Danish physiologist, well known. He came up with this sentence, which has been called as the Krog's dictum. And in his eyes, studying animals, any kind of species, can be interesting for many interesting reasons. Not only because it's interesting to know how they work, but also because they can help us solve other problems. For example, he explains the case of his teacher. Christian Bohr was a well-known, also Danish physiologist. And Christian Bohr was interested in the respiratory mechanism of the lung. What Christian Bohr used was actually a turtle species in which the trachea divides high up in the neck. Therefore, every single lung actually is connected by a long bronchi. And he's saying, well, this animal is particularly suited to study this because then you can actually compare the response in one lung to the other, and you can do that because the bronchi is very long and you can study them separately. This gives August Krog basically gives us this as an example. Why I'm bringing this up? I'm bringing this up because what have we seen in rats? 2 days. 2 days is the time you have to study this. How much, or what's the difference in penguins? Fasting lasts much longer. So why not study penguins? And actually, a lot of studies on fasting animals have been done in penguins. Perhaps it's one of the animals which we know best, this response. And the fasting period has been divided in 4 phases. Well, this is my making. If you read the literature, they will tell you 3 phases. But implicitly, there is the first phase, which I call phase 0, which is the pre-fasting. And I think it's important to highlight this. Without this phase, the other phases will not take place or will be much worse. The phase 0 that I call it is the stock up on fat and proteins as reservoir. what animals, in this case penguins, do is before they go on a fast, they put on a lot of weight in the form of fat and also proteins. In male emperor penguins, 30% of the body mass, 30% of the body mass is fat before starting incubation. And of course, this is extremely important. If the animal does not put enough fat, it's not going to be able to endure the 4 months of the egg incubation in the winter. Phase 0. Phase 1. This is a rapid adaptation phase in which the animal, it's the very beginning of fasting, when the animal starts fasting. There is a reduction in the loss of body mass, and there is the start of fat mobilization. This phase is very short, and it's hard to study, at least in penguins, because it takes place when the animals are still returning to land. It's known that in penguins, when they are getting back on land, they are already fasting. They are already in what's called phase 2. That's the economy phase. In the economy phase, there is a low rate of loss of body mass, and there is also a low contribution of proteins to the energy state. During phase 2, what happens is that the animals are using up fat, are using up lipids. Not only that, but there is also a reduction in metabolic rate. So during phase 2, reduction in metabolic rate and start using lipids. With some protein, unavoidable, as we will see in a second. This phase can be 52 days instead of 48 hours in rats. So that makes it much more easier to study. Finally, phase 3, that's the phase of muscle breakdown. In the muscle breakdown, basically the animal starts using protein. And which protein? Protein from the muscles, wasting muscle. Fat stores in this phase are not completely exhausted. There's always some fat left, but it's more and more that proteins are used. And you will see in the next slides how we can recognize these phases. As I said, loss of body mass, what you see here in the different phases. During phase 2 conservation, of course the animals are losing weight, but not at the same rate. What you have here now is actually the use of protein versus the use of lipids. Lipids are being used during phase 2, and then there is a transition to phase 3 when lipids start being less used because we are getting close to 0, and when the use of protein actually increases. What's important for you to understand is that protein have something that lipids don't have. Proteins have amino acids. Amino acids can be converted via gluconeogenesis to glucose. So it's unavoidable that some protein is lost for the glucose that is needed. That cannot be provided by lipids. But once the energy provided by lipids is almost gone, then proteins kick in. And this is the dangerous phase in a fasting response. And as you will see it later in another lecture when we discuss the penguins. The data that we have for penguins tell us, for example, that there is an increased circulation of fatty acids. The fatty acids actually increase in the circulation together with the ketone bodies in phase 2. Why? We are breaking down fat, nothing else. Amino acids, in this case alanine concentration. Well, there is a certain level here. Why alanine? Alanine is one of the gluconeogenic amino acids. Alanine can be converted into glucose. The levels remain, they start being elevated close to the end of phase 2, and then they increase more markedly. Urea concentration. Urea is actually an indicator, urea and uric acid are indicators for protein metabolism. Notice the remarkable increase during phase 3. Both of them shoot up. During phase 2, there is protein metabolism, we have seen that, but at a much lower rate. During phase 3, energy is provided by protein. Gluconeogenesis actually takes place. That's the way to support energy, and muscle wasting is happening. Essentially, we know as well that in this interface, and actually you could see that here, proteins are being used at all times. So, Protein catabolism anticipates the total loss of lipids. But when proteins are lost, or too much of them, it's basically regarded that loss of 50% of proteins is regarded as lethal. So when the animal is pushed to this end, things are getting on thin ice, so to speak. The animals basically are no longer able to get nutrients. There's very little fat reserves. Protein is actually being wasted using for energy, not the main purpose for it. And what we see is that when we correlate this with corticosterone, the main glucocorticoid, we see a dramatic increase in phase 3. So we regard corticosterone as the best marker for phase 3 fasting. Corticosterone during this phase actually promotes protein breakdown and promotes gluconeogenesis. As a summary here, this is what you see, essentially, corticosterone increasing in phase 3. Plasma hydroxybutyrate. This is an indication of fat metabolism, kept high during phase 2, falling by phase 3 because there are fewer fat. Uric acid increasing in phase 3 because of the protein metabolism. And notice the loss of body mass. The animal is losing body mass at a given rate here, at a steady rate. So there is a loss, but it's steady. On the onset of phase 3, that basically goes up. And this is when things change for fasting. So I would sustain that phase 2 is a natural, is a physiological fasting state. In phase 2, the animal can actually handle energy properly by burning down fat. When it comes to phase 3, basically proteins are being broken down, and this would be a pathological situation, a pathological state of fasting. As an example, when people go on a hunger strike, what is the main marker for doctors to say, now this person is reaching the limit? The production or the presence of urea, of nitrogenous compounds in the urine. When nitrogenous compounds in the urine increase, it's an indication that this person is breaking down proteins, therefore that is entering this phase 3. I would say that most animals, we humans, are not different from these responses. It's simply that these responses happen faster. Earlier on, you will see these changes earlier on. That's basically it. And that's the end of this lecture. Thank you very much.